Hello, my name is Julie Bowles, and today I will be discussing Case Study 2, the Prenatal Microarray Screening. The case study I selected covers a couple, Janelle and Spencer, who are wanting to conceive a child and are wanting to determine if they carry any autosomal recessive conditions. In each gene that is associated with the disease, a DNA microanalysis can be conducted. First, a sample of DNA is taken via a blood sample. The DNA is then separated from two complementary strands into two single-stranded molecules. The DNA is then split into fragments and stained with a fluorescent dye. The control or normal DNA is labeled green, while the mutated DNA is labeled with red. The dyed DNA is then bound to the synthetic DNA on the chip. If the individual does not possess a mutation, both samples will bind to the normal DNA. If the individual does possess the mutation, the red sample will bind to the mutated DNA. Here's a picture depicting how this process works. I chose this particular case because my husband and I are also trying to conceive a child, and I'm interested in genetic testing. While we are both of European descent, I'm curious to know if we are carriers for any autosomal recessive traits as well, and how that would impact our future children or would have any effect on our decisions regarding what to do if they were affected or had the potential to be. The first question posed in this exercise was, why is it important to know the penetrance of the illnesses? Penetrance is the magnitude of which a particular gene or group of genes is expressed in the phenotypes of individuals who are carriers for it. It can be complete or incomplete, in which an individual possesses the genotype but may not be fully expressed. The importance of knowing the penetrance of the illnesses is to determine individuals who are carriers for a particular genetic mutation, assist in decision regarding reproduction, assess family history, and identify early on newborns who would benefit from early intervention, depending on the type of genetic trait. In the case study, it is apparent that both Janelle and Spencer are not carriers for cystic fibrosis or osteogenesis imperfecta. However, Janelle and Spencer are homozygous recessive for beta thalassemia and albinism. Janelle is homozygous recessive for adenosine deaminase deficiency, and Spencer is homozygous recessive for hemochromatosis. The second question, Janelle is an African American and Spencer is a Caucasian of English heritage. Why might the particular disease causing alleles in their microarrays be different? SNPs, single nucleotide polymorphisms, SNPs are a common genetic marker. They are variations that occur when a single nucleotide in a DNA sequence is altered via a base pair substitution, in which the original or ancestral allele is replaced with a new or minor allele. While SNPs are common, mutations in an SNP are rare. But are common throughout generations. Diseases can be traced to specific genetic markers. Certain populations may have a higher incidences, like in African American women, where hypertension, sickle cell anemia, melanoma, breast cancer, and sarcoidosis have a much higher incidence than in Caucasians. And in English heritage, hemochromatosis, cystic fibrosis, lactase persistence, celiac disease, galactosemia, and multiple sclerosis have a higher significance. The third question is, what information about the interactions of alleles is important to know to interpret the results of this test? Genetic testing may be able to identify a mutation in a gene or chromosome and confirm if the person is a carrier. If there's an increased risk of the fetus developing a disease depending on penetrance, or to confirm a diagnosis. However, a positive result for a predictive test cannot establish the exact risk of developing the disorder or the severity of the condition. While a negative result may be able to give some peace of mind, genetic testing is not 100% accurate, and there is always a possibility that the test could miss a condition and provide a false positive. Therefore, it is important to know the percentage for each condition and the likelihood of the child developing that condition. To interpret the results, it is important to remember that the incidence of their offspring developing one of the conditions is low. The fourth question is, why is a DNA microarray test for an autosomal dominant condition with complete penetrance unnecessary? The reason for a DNA microarray test is to determine if the parents are carriers of the condition and ultimately if their child will have the condition. But if both parents are autosomal dominant, there is no need to conduct the test. Complete penetrance in a dominant gene would be 100% present in all offspring. The fifth question is, which disorders does a child of Janelle and Spencer face a 1 in 4 chance of developing? Janelle and Spencer are both homozygous recessive for beta thalassemia and albinism, which is a 1 in 4 or 25% chance that the child will receive 2 normal copies of the gene and will not be a carrier and will also be asymptomatic. And then for the adenosine deaminase deficiency and for hemochromatosis, in which Spencer and Janelle are singularly homozygous recessive, they will also have a 1 in 4 chance of developing the condition. Number 6, the probability that their child will be a carrier for hemochromatosis is, in this case, Spencer is homozygous recessive for hemochromatosis. The child would be asymptomatic, but would still have a 50% chance of being a carrier for the condition. And in the chance that a child of theirs would be a carrier for cystic fibrosis and a carrier of adenosine deaminase deficiency would be very low, 0%, because they both don't carry that condition, that disease. And lastly, the final question posed was, explain how a false negative can result from the test that these two people undergo. Microarray DNA technology can predict the likelihood that one or both parents are carriers for a trait that can be passed on. The issue arises in the amount of genes that are being interpreted, since there are a significant number that are analyzed simultaneously. The false negative rate can be determined by 4 factors: measurement variability, sample size, distribution of differences, and proportion of expressed genes. Another study found that variability amongst data interpretations, specifically due to the fluorescent images being interpreted by computer data instead of verifying the raw data, was another source of false negatives. While the technology has been around since the 1980s, it is still a work in progress as we continue to understand and interpret how chromosomes and genes work. In regards to the relevance to the class, the case study makes what we're currently learning in the class is primarily located in chapters 35 through 40 in terms of SNPs and genetic markers. Chapter 40 covers population genetics, which we discussed to determine the difference between both populations of racial groups and the associated diseases that can be found in higher prevalence with ancient alleles. A microarray DNA analysis covered in chapter 37 is used to determine prenatal conditions by isolating normal and mutant alleles and how they can determine if 2 parents are carriers, heterozygous or homozygous recessive for particular traits. And this concludes my presentation. These are my works cited pages. Thank you.